Okay, we're not going to go through and talk about each of the individual classes of macromolecules in more detail one at a time. We'll start out with the carbohydrates. So typically, when you think of the word carbohydrates, you might think of nutrition and eating a lot of carbs when you need a lot of energy. And so carbs typically are going to include sugars, like what we usually would think of as a sugar like sucrose, table sugar. But this also includes the polymers of sugars, things like starch, right? Starchy foods have a lot of carbohydrate in them. Carbohydrates can be of a variety of different sizes, and so we can divide them up essentially into 3 different categories. Each category, the name for each category has the term saccharide in it, which means sugar, and we give it a prefix. So for example, monosaccharide would be your simple or your single sugars. Then we have your di for 2, disaccharide. These are going to be your double sugars. And then lastly, we have polysaccharides. Again, poly meaning many, and these are going to be the ones that are considered the polymers or those carbohydrate macromolecules. Really, really large carbohydrate molecules that are put together essentially by just stitching together a whole bunch of monosaccharides together. So let's talk about each one of these subcategories of carbohydrates, again starting with monosaccharides. So monosaccharides are going to be your simple or single sugars, and they typically have the molecular formula that's a multiple of CH2O. For example, glucose is going to have the molecular formula C6H12O6, and that is one of the most common monosaccharides. We're going to spend a lot of time talking about glucose, both in this chapter, and then also as we start our discussion of cellular respiration later on. There are many different kinds of monosaccharides, not just glucose, and we can categorize the monosaccharides based on essentially 2 components. Number 1, remember that sugars tend to have the carbonyl functional group in it. Remember that the carbonyl functional group is essentially a double bonded oxygen to carbon within the carbon skeleton of the organic molecule. And one of the unique features of the carbonyl group is that depending on where you put that double bonded oxygen, whether it's in the middle of the carbon skeleton or at the end of the carbon skeleton, you can essentially create 2 different types of molecules. So if you take a look at the table on this slide, and you take a look at the columns, the column on the left, these are going to be your aldehyde sugars, or your aldoses. Now, aldehyde sugars are going to have that carbonyl group attached to the end of the carbon skeleton. On the other hand, on the column on the right, you have your ketone sugars, or your ketoses. These are going to have that carbonyl group that's somewhere in the middle of the carbon skeleton. So, location of the carbonyl group is one way to categorize those monosaccharides. Another way that we can categorize them is based on how many carbons that they have in that carbon skeleton. So for example, now going and looking at the same table, but just by row instead of column, in the first row we have our trioses. These are going to be our 3 carbon sugars. Things like glyceraldehyde or dihydroxyacetone. We're going to talk about glyceraldehyde again when we go back and take a look at cellular respiration later on. In the middle, we have our pentoses. These are going to be our 5 carbon sugars. A really important 5 carbon sugar is going to be our ribose. Ribose is actually one of the components of the RNA building block. We'll talk about those in a little bit. And then lastly, on the bottom, we have our hexoses. These are going to be our 6 carbon sugars. This is typically what we would think of when we hear the term monosaccharide. So your glucose, your galactose, your fructose. Now, when we typically think of glucose and we write it out, as you saw in the previous slide, we write out a linear structure to the carbon skeleton for glucose. Keep in mind that when we add glucose to an aqueous solution, it tends to form a ring-like structure. So if you take a look on the top part of this slide, you'll notice that the first carbon 1 here, is highlighted in pink, and then the 5th carbon, the hydroxyl group there in the 5th carbon, is highlighted in blue. And these are going to be the 2 parts that are going to help in the ring formation of the glucose molecule. So if you take a look, that hydroxyl group from the 5th carbon, that oxygen is going to form a covalent bond with the carbon that's in the first position within the glucose structure, and that will form this lovely 6-membered ring and completing the ring structure for the glucose molecule. Now, sometimes you might see that full structure that's in the upper right-hand corner here for the glucose. Sometimes we like to abbreviate the ring structure, which you can see in the lower left-hand corner down here. When you look at that abbreviated ring structure, essentially all we've done is we've eliminated the carbons within the ring, and we've just sort of implied them. They're still numbered in this case. You can imagine there's a carbon at each one of the vertices of the hexagon. If it's not a carbon, like you see that oxygen between the fifth and the first carbon, will actually tell you what that atom is. The other thing you'll notice about the abbreviated structure is that it has that fat line on the bottom. And this is essentially trying to show you that that ring structure is actually three-dimensional. It's hard to do this when you're looking at it on a two-dimensional medium like a screen. But if you imagine taking that ring structure and pulling it out of the screen, you would essentially lay the ring parallel to the floor, and some of those components that are attached to the carbons, either the hydrogens or the hydroxyl groups, they're going to find themselves either on the top of the ring or on the bottom of the ring, giving that three-dimensional shape to the molecule. And as we'll see in a little bit, it matters whether those components are on the top or on the bottom of the ring. The other thing it's important to note is that while typically we consider monosaccharides as a really good source of fuel, like glucose, for your cells, cells are really good at reusing materials. They don't like to start from scratch, right? They'd like to take the easiest, shortest route possible to get to wherever they want to go. And so having that nice 6-carbon skeleton in monosaccharides like glucose, that serves as a great, essentially raw material, right? It's a good start for building other things. You can modify a couple of the functional groups, maybe add some more carbons, and now you have a completely different molecule, and you didn't have to start from scratch. So while yes, monosaccharides are great as a fuel source for cells, but they're also very important to serving as this raw material for building other molecules. Now, we don't just have to stop with our monosaccharides. For example, we can take 2 monosaccharides and stitch them together. When we do this, we end up with a double saccharide or a disaccharide. And the way that we stitch them together is by using a dehydration synthesis reaction, which we talked about previously in the introduction for chapter 5. When we stitch those 2 monosaccharides together, we're essentially forming a covalent bond between those 2 monosaccharides. And we give a very special name to this covalent bond that stitches monomers for carbohydrates together, and that's a glycosidic linkage. So on this slide, you can see that we're trying to form the disaccharide sucrose. Now, sucrose can be formed by taking 1 glucose molecule and attaching it to 1 fructose molecule. Again, this is a dehydration synthesis reaction, so we have to remove a water molecule. You can see how we can do that by removing parts of the hydroxyl groups from glucose and fructose. Water molecule comes out, and we end up then linking the oxygen to that first carbon on glucose. And this forms the glycosidic linkage. In particular, this type of glycosidic linkage, which links the first carbon and the second carbon of the adjacent monosaccharides, this is called a 1,2 glycosidic linkage. Not important to memorize the numbers, but just when you see them, you know where that term comes from. It's essentially labeling those carbons involved in that particular glycosidic linkage. Okay, we don't have to just stop at disaccharides. We could also continue on to polysaccharides as well. And so for polysaccharides, we're essentially taking multiple monomers or multiple monosaccharides of the carbohydrates and stringing them all together. Right? Now, polysaccharides can have 2 different kinds of roles. They can either have a structural role or they can have a storage role. And what determines whether you're going to have a structural or a storage polysaccharide is essentially how that particular polysaccharide has been put together. So what types of sugar monomers were used, and then what are the positions of those glycosidic linkages that hold the monomers together. So let's take a look at examples of both structural and storage polysaccharides. Storage polysaccharides first. So animals and plants use 2 different kinds of storage polysaccharides. Plants will store all of their excess energy in the form of starch. So let's say it's a nice sunny day out and a plant is performing lots of photosynthesis. It's probably going to produce more glucose than it knows what to do with at that given moment. So it's going to try to store some of it for later, maybe on a rainy day when it's kind of cloudy, or maybe at night, right? Plants don't die at night, even though they still need energy to survive. They just turn to their energy reserves, aka their starch molecules. Now, starch is essentially just made out of glucose molecules that are all strung together to form that polysaccharide. If you take a look at the top picture, you'll notice that amylose is one of the simplest types of starch, and it's really just a long chain of those glucose molecules all attached to each other. And it also has sort of this coil-like shape to the molecule, so it's not straight, but it's coiled. Another example of a type of starch is your amylopectin. Amylopectin is a little bit more branched, but still the same idea applies. It's all just made out of glucose molecules and has that coiled shape to it. Now, we as animals, we don't use starch to store excess fuel. We use glycogen. So remember, you eat that high sugar meal, your blood glucose goes through the roof. Your pancreas secretes insulin and tells both your liver and your muscle cells to take up some of that extra blood glucose. What do they do with it? Well, they build glycogen. So your liver and your muscle cells will build glycogen, essentially taking all those individual glucose molecules, and through the process of dehydration synthesis, they will link them all together to form glycogen molecules. So glycogen is very similar to starch, and it's just a long string of those glucose molecules, except glycogen can be a lot more branched than your starch molecules can. And then, let's say you haven't eaten in a while, and your body still needs that fuel to get through to your next meal. It's depleted its blood glucose, so then it turns to its glycogen stores in the liver. It says, hey liver, we need more glucose. So your liver then goes through the process of hydrolysis to break off one glucose molecule at a time to release it then into the bloodstream to feed the rest of your body's cells. Okay, the other kind of polysaccharide you can have is a structural polysaccharide. And that you can have it, but really plants have it. So when it comes to structure, you have a skeleton. It gives you support. It allows you to stand upright against gravity. Plants don't have a skeleton. So instead, they have a molecule called cellulose. Cellulose, again, is a polysaccharide, and it's one of the main components of these really rigid plant cell walls. Now, this isn't something that we've talked about yet. Once we get to chapter 6, we will, as we start introducing cell structures. But essentially, this is one of the main defining differences between animal cells and plant cells. Plant cells are going to have these tough cell walls on the very outside of their plasma membrane, whereas animal cells don't have these. And just like starch, or the storage polysaccharide in plants, cellulose is also made out of glucose. So why are these two molecules different if they're both made out of the same monomers? Well, it comes down to those glycosidic linkages. How are the glucose molecules linked together? It's different in cellulose versus starch. And the two differences really come from the fact that glucose, when it makes that ring structure in aqueous solution, it can do so one of two different ways. It can form either the alpha ring form or the beta ring form of glucose. The starch is going to end up using the alpha ring form, and the cellulose will end up using the beta ring form. And this creates a lot of difference both in the structure of the molecule of the polysaccharide as well as the function. So let's take a look then at how cellulose is actually constructed and how that's different from starch. And remember, cellulose is going to be found in that tough cell wall that is on the outside of plant cells and helps give them that rigid extra support and protection.